Rock Springs High School students. We are so lucky today to be joining Brett Ralston from his research lab. Now, some of you actually already know Brett because he was there that auspicious day in Rock Springs High School when we brought clay and you all dropped balls of clay and measured clay plasticity, and we talked about microbes in clay. So you may remember Brett, but now we get to see him in his native habitat where he does all of his research, probably spends lots of hours every day in the lab. So Brett, tell us broadly a little bit about what you study.
I study reproductive physiology, which is this idea of how reproduction works in the system of a living organism, so in the system of humans specifically. And so I'm looking at the pituitary, which is right in front of your midbrain. If you're familiar with anatomy of the brain, you might be learning about it in class right now. And it releases hormones, and it's the cause of all the puberty problems that are mentioned in health class that you might be embarrassed to talk about. But I look at the cells, two cells in there called gonadotrope cells, which is just the name for cells that release hormones that act on the reproductive organs in males and females. And then I'm going to be looking at lactotropes, which is actually important for releasing something called prolactin, which is important for making sure that pregnant women can, one of the things is help them produce milk for their for babies.
Yeah. So like these chemicals are released from the pituitary and then they travel through your body and they target particular organs.
Yeah. And that it's all just a giant regulation system. So those go and target specific tissues, and those tissues release chemicals that go back and regulate the pituitary gland. So it's this cycle of maintaining homeostasis or balance in the body.
Cool. So we could think about it like as sort of a really good conversation where it's each participant is playing a role. So it's not just your brain talking to your body, but it's your body talking to your brain.
Yeah, that's a great way to put it. I guess I also forgot to mention that the other aspect that I look at in relation to that is how those hormones are regulated at the genetic level. So yeah.
Yeah. So the students have been talking about genetics, but one thing I'm not sure they've talked very much about is epigenetics. What is the difference between genetics and epigenetics?
Yeah, so I would describe the difference between the two is genetics are being like what your DNA is specifically coded and what that says. So it's like the ACTs and Gs, how do they get turned into proteins, and what do we typically think about how those genes are turned into proteins? Whereas epigenetics is more how the DNA is read and how it's read changes based on the environment. So, for example, and this is smoking and like obesity can actually affect how your genes are read, which can change the way that those proteins are created, which in effect changes how your body responds to certain things.
Yeah, that's really interesting. I was just trying to think of an analogy in my mind, like this idea that, if you're reading a book, the words are always the same, but if you're reading outside under a tree on a beautiful day, your response to that reading might be different. Like it might change how you respond to it. So it's like, the genes are always the same, but like how those genes are expressed or how they get read at any given time kind of changes.
Yeah, exactly. It's just it's how the genes get interpreted differently under different conditions.
Yeah, it's super cool.
Yeah.
Super cool. Yeah, so Brett, show us a little bit of the nuts and bolts, like what's on your benchtop that helps you do what you do?
Yeah. So I'm sitting at my benchtop right now. It's pretty clean. I like to keep it clean and organized. These are my pipette tips for measuring out small quantities of solutions. This is my trusty lab notebook where I ramble on, and this is a new one, so it's pretty fresh, where I ramble on about what I did in the day and what I'm going to do in other days. And then I started an experiment today. So is the sound quality okay in here?
Yeah, you sound great.
Okay, so right next to my lab bench is this really cool machine called a PCR machine. And here is actually it in the progress of working. See, it says it says stop run, which means it's running. It is on. There's still an hour and a half remaining. But what this does is this allows me to see like amounts of the DNA that are in the cells and organisms that I'm looking at by taking cells from them and then extracting out the DNA. You might have done like a DNA extraction in strawberries at some point, if that's familiar. And you can run a PCR on it to amplify a certain gene that you want and to see how much that gene is present or to see how much DNA is there representing that protein.
Awesome. So essentially that PCR instrument actually makes like more and more and more and more and more copies of the DNA or the gene you're looking at or whatever region of the DNA you're curious about.
Exactly. And yeah, that's what this increase here is showing. And it just moved actually, which is really cool. I've never seen that.
That's so cool.
It's showing an increase in it's amplifying the DNA, and it's showing the ones that usually come up. Sorry, let me explain this better. So it runs a certain number of cycles, which is how many times it just repeats the process. And ones that come up earlier show more DNA because there was more there, so it should amplify faster because it's an exponential increase in DNA.
Awesome. So what are you amplifying? Like what gene are you making more copies of right now? Or
yeah, so to go a little bit more specifically into what I do, it's actually I'm replicating a gene called DGCR8, which just a physiological jargon term. We like to name our things with letters and numbers and make it difficult sometimes, but it's important for the processing of other regulators like microRNAs specifically. So if you've heard about microRNAs, they are short strains of RNA, which RNA is it goes the central dogma DNA to RNA. It's the same RNA that's there and then transcription, but these RNAs do not get turned into proteins. These RNAs go and actually silence RNAs that are going to make proteins. So it's a way of this protein regulates the regulators of our proteins. It's kind of, I have a lot of up and down in what I'm doing. So.
Yeah. So why is that? Why are the regulators like if we know that genes can get turned on and turned off and that regulates gene expression, like why does a biological system want to regulate the RNAs too? Like it's like a whole nother way to regulate. It's like, oops, I forgot to regulate the DNA. Now I have to regulate the RNA. Like what led to that?
So I don't know. Evolutionarily, I'm not exactly sure all the pathways that got us to all these methods of regulation, but it seems to be that regulation is kind of the name of the game when it comes to like maintaining balance in our bodies. So there's ways to regulate proteins by maybe marking them with little groups that your body goes and destroys them. Or we can regulate the RNA, which would get turned into the protein. So before the protein even gets turned into it, we can stop it from being made. And there's other methods of regulation. However, what's really cool is you can also, if you want to not see a decrease in those, you can regulate the regulators to show them increased. So it's this complex, really cool system of things stopping each other from working and then letting them work again. And then it's just this balance that goes kind of in a cyclical fashion to maintain a balance in your body.
Yeah. I mean, I guess that makes sense, but when you think about like how we regulate traffic, it's not just one thing. It's like we have a speed limit, but we also have a side of the road that you're supposed to drive on, and that regulates traffic. And we have stoplights, and we have roundabouts, and we have like all these different levels of regulation. So it seems like it makes sense that our body would also like regulate here and then regulate there, and just give all the possibilities of letting everything flow smoothly, just like traffic flowing smoothly within your body.
Yeah, I really like that analogy. That's good. It's interesting just to think in general about the kind of ideas that we get from life and how we apply them to infrastructure in the world around us. And, maybe that didn't come from thinking about regulation in our body. But there's definitely things like somebody modeled a plane after a bird that they saw because they noticed how aerodynamic it was. So it's kind of cool how life found a way, and sometimes we mimic it. Biomimicry, I think that's what it's called, actually.
Yeah, super cool stuff. So go back for a minute, because you said the gene that you are making more copies of or amplifying is DG, help me.
DGCR8. It stands for DeGeorge syndrome chromosomal region 8. So it's talking specifically, it was named after the George syndrome disease, and it was found to be located within the region 8 of, I don't remember which specific chromosome it's on. So that's just how it gets its name and why we classify it that way. It has to do with the disease that we observed and then went back and found the where it went wrong in the genome. So we're looking at it because at least a hypothesis that I have is that it helps regulate microRNAs that might be responsible for releasing those hormones that I talked about that were released in the pituitary. So so there's many different ways. There's usually more than one idea that you can extrapolate on when it comes to one thing that you're looking at, and that there's so many different answers, but there's a focus kind of the lab that I'm in.
So show us what you would do after you amplify your DNA. Like, what's the next step? What would you do with it?
Yeah, so do you want to see the data? I can show you some of the data. And this isn't going to make any sense probably, but this is just kind of what it looks like. So just a bunch of numbers, which is what we get to deal with. Essentially, this might help. This is my sample name. And so this is the 21st sample that I've made for this experiment. This sample was this is my control, and this is my one that I was treating with the experiment. And so essentially these numbers are just what the PCR machine gives out, and then this number at the end is how they differ from each other. So I have my control, and then I have my experiment. So these ones didn't change that much. The DGC, so this is looking at DGCR8 levels. See how they're both like 1? They're both around 1. But this is kind of exciting. These two, this is my control and this is my experimental. So DGCR8 under this condition dropped by about 60%, which is really cool. And that's kind of what we're looking for, because actually in that experiment, I knocked down DGCR8 with something similar to a microRNA, but it's created by us. It's called an siRNA. So it's we made it and we put it in the cells, and the cells uptake it, and it goes and silences the genes like the microRNAs would, but we do it for experimental purposes.
So that you can.
Sorry, that's a lot.
What it does, yeah. So like a lot of times researchers do that. They turn a gene off or they silence it so that you can see what it is that it's doing, like what changes when it's no longer being expressed. So yeah, that makes a lot of sense. And Brett, do you work further with the DNA once you have amplified it?
Yeah. So I'll first run a PCR on DGCR8 to see if it's knocked down, because it should get knocked down when we silence it with the siRNA. And then if it worked, I take that DNA and I run to see if those microRNAs that I was talking about are changed when DGCR8 is changed. And then I take the same DNA and I also run the downstream targets on that PCR to see if there's a change in those suggested targets. So essentially, I'm trying to see if when DGCR8 is knocked down, are these targets actually being affected that might have something to do with the release of hormones, that might have something to do with the dysregulation in the pituitary, which might lead to pituitary cancers if it's unregulated, because the cells can just proliferate a lot and not be controlled. So that would be cool to uncover something there. And so it's kind of a process of using this DNA under multiple circumstances, quality checking it, and then going back and doing another experiment with it.
Yeah, so show us a bit more on the benchtop what you use to do that.
Yeah. So this is a kit that we can use. This is a RNA extraction kit. So this allows us to take the cells that we have and lyse them, or pretty much just blow them up and get their nucleic acid, so their DNA, RNA. And so it has these bottles of buffers in them, and they all have different names, and they all are important for different steps in the process. And there's an instructions thing and tubes that you can get your RNA into once you've done the whole kit. And then you can take that RNA and do reverse transcript. Oh man, we take the RNA, and then we actually, next to this beast PCR machine is this tinier machine, which it can do reverse transcriptase PCR, which takes RNA and converts it back into DNA, which is really cool and useful tool. It's actually been used quite a lot for like the genomics project and figuring out coding sequences of DNA. So after we do this kit, this RNA extraction, and after we do the smaller machine PCR to get the DNA, we then get to take that DNA, and I wish I could have had the PCR. I should have been doing it in the middle of this. That'd have been cool. But there's other reagents in this freezer, and so we have to get a bunch of reagents together. So this is a reagent that helps run the PCR, the big machine PCR, and it has to be kept in cold conditions because we don't want those. I'll show you one second. I'll explain it in a second. These are kind of, I would say, the really important part of PCR and how you choose what you're amplifying. So these are my primers. And what primers are short sequences of DNA that bind to the cDNA that you're looking at, and then you can amplify it in the PCR. So this is my DGCR8 primer. So I'd use this. I'd mix it with my sample that I got. I'd mix it with that other green box, and then I could put it in a 96 well plate, which I'll show you. One of these plates, which is called a 96 well plate, because there's 8 letters, or that's how many wells there are on the plate. And so I'd put my DNA and my kind of mix of all those reagents that I was showing in here, and then it fits into this slot over here on the PCR machine. So that would sit in. This comes up, and it hits the top of the PCR plate reader, and it goes through these stages. It's on stage 2 of 37, or 37 of 40, and that's when I get the data out here. And then I would do the stats and calculations on it that I showed you on my computer. I know that was a long process.
Yeah, perfect. Thank you. Yeah, that's awesome. And
I would like to do that with people in the lab. That'd be kind of fun. I don't know, to run a PCR with you guys.
Yeah, I mean, next time, hopefully that's what we'll be doing, is visiting in person, right?
Yeah, that'd be cool.
And we'll do PCR with you.
That'd be fun, yeah. qPCR is it's a really amazing, useful technique, I think.
Yeah, so PCR is probably arguably the most important technology of our era, because it's allowed us to amplify DNA for purposes of identification, but also to do it for purposes of experimentation like you're doing. So anything from crime scene investigation, where you're trying to make copies to identify who a perpetrator might be, all the way to doing things like you're doing to really hone in on the function of a particular gene. So there's just everything in between. And I know Sydney does a lot with that too. Do you want to take just, if you don't, Sydney lives in a place where there's loud planes. So if you have loud planes, you can just say no, but what are you using PCR for?
Yeah, so I recently worked at the Wyoming Public Health Lab this summer helping on the COVID surge team. So we use PCR when we get samples. So if you go get your nasopharyngeal swab, if you think you have COVID, they send that sample to us. And then just like Brett showed, we remove the nucleic acid and then run it on PCR to see if there is the SARS-CoV-2 virus detected in those samples. So it's a really common technique. I also use it in the research lab I work at the university, and we're looking at iron regulation and immune responses. So as Rachel mentioned, probably the most important, it's just so diverse in its uses. So
yeah, in fact, I don't even know. I looked up at one point the multi-billion-dollar industry of PCR. I mean, it's absolutely huge. It's just insane, and really it's a game changer for science. To think that like 1983 was when that was discovered. So it's, from that time to now, how much it has changed in like being able to do it like in the thermocycler that you're showing, that's like really high tech, from when Kerry Mullis invented it, where he was putting the DNA into different water baths and literally denaturing it and allowing the primers to anneal, elongating, right? All these different temperatures. And now, of course, the machine does it for you. That I shouldn't call it a machine. Instrumentation is the more proper term, and the instrument. So thank you, Brett. What do you think what's your favorite thing about going to work each day? What do you like the most about working in the lab?
Oh, I would definitely say asking questions. Like, I get to do, what is the lab's purpose, and then I get to expand on what we know and try to like extrapolate ideas. And I just like thinking about what the implications are of our research. And that's kind of my driving, factor. I like to try to do the problem solving. That's probably that's definitely my favorite part when I come to lab and work with all my other lab mates. I don't know. I don't know how to explain that feeling though.
It's just good being able to be curious.
Yeah,
I'm thinking about the Mr. Rogers video that they made. Like, I think when he died, right? It's great to be curious about a great many things. So I think curiosity is the best thing we can have, and a job that lets us be curious every day. It's a job of life, right? As opposed to feeling like you're dying a little more each day when you go to work. So, having that capacity to be curious every day is huge.
Yeah, it is. It's definitely also, I would say, a privilege in some ways. So.
Yes, very much a privilege. Yeah, and I think it's just wonderful that you're grateful for it. Yeah. All right. Well, thank you.
Yeah, it was fun. It was great to have you.
Yeah. And next time it'll be in person, right?
I hope so.
So Rock Springs High School kids, you should email us or make a Google Doc with questions so we can get back to you with questions. And I know Brett would be very excited to answer any questions that you might have.
It'd probably be less confusing through an email. Hopefully.
Email or Google Doc or whatever works on your side is totally fine.